So let's launch straight into today's program. And it's a terrific privilege for me to be able to introduce Professor Ranjeny Thomas, who is a fellow of the Academy, a former chair of the Queensland chapter of the Academy, and a member of the Order of Australia. Ranjeny is Professor of Rheumatology at the University of Queensland and holds a position also at the Translational Research Institute. She's a consultant rheumatologist, and as I've just said, has many distinguished awards. In addition, she is achieving the holy grail, if you like, in that her research is actually translating directly into patient benefit. She founded the UniQuest spin-off company Dendritic, which seeks to understand autoimmune disease and the restoration of immune tolerance. Ranjeny has a stellar track record, immense amounts of funding, accolades galore. So it's going to be a great privilege and pleasure for me to introduce Ranjeny. Ranjeny, the floor is yours. Thank you.
If you could stop sharing, I can actually share.
Oh yes, now that's going to be the trick, isn't it? Let's do that. Stop share. The technology. Okay, I'll just.
Oh, good. All right, good morning, everybody. So I'm sitting at a very interesting interface for the clinician-scientist, translating innovative therapies to the clinic, the interface of science, being a doctor, and also interfacing with industry. And I'll tell you about that journey today. So my translational journey started with my basic physician training, and during that I got interested in immunology through our journal club, and I started advanced training in rheumatology, and I was training in WA, got involved in some clinical research projects, and they really leveraged my interest in immunology and introduced me to flow cytometry. And to the extent that when I finished, when I was getting close to finishing my advanced training, during my non-core year, I decided to start my doctorate, and I moved to University of Texas Southwestern in Dallas, and I decided to see how I would go with basic immunology, and my project was in basic human cellular immunology. So once I'd finished that, and that was in the basic biology of dendritic cells, I was Queensland. The position is supported by Arthritis Queensland, the peak consumer body, and it's to do research, teaching, and clinical. And how I started off was in basic research, because that's what I knew, in antigen presentation, but I moved it into learning about rheumatoid arthritis patients. And I then started expanding into preclinical mouse work, in vivo immunology, and looking at transcription factors by recruiting people around me who, and collaborating with people that knew about those aspects. I never actually did a postdoc. I sort of learned it on the job. And so by 2003, we published a paper. We're able to then start translating a basic science discovery that was published in 2003. And that led on to a proof of concept clinical trial, which we started in 2011. And by July 2013, and I'll tell you about the story as well later, we got some investment from the pharmaceutical company Janssen for preclinical development of a novel therapy, and that was all the way through to clinical trials. And they took an option to license our innovative drug in rheumatoid arthritis. And then in 2017, we started collaborating with 2 further pharmaceutical companies on immunotherapy. And also then we were awarded a US philanthropic grant for preclinical development and a clinical trial in type 1 diabetes. And since 2017 to 2019, we carried out the clinical trial in RA patients. And Q1 of 2023, we start our clinical trial in type 1 diabetes. So here's the cycle of translational research, and it really starts with a research question. And this is T1, or translation part 1. And you're looking at your fundamental science and preclinical research to really see whether your idea is tractable to be translated. And at that stage, you might make a discovery that could be patented, and then start to do some proof of concept work to see if that's really translatable to something that could be developed. And at that point, you need funding to be able to develop it commercially and get enough money to develop it in a larger scale. And if you get through that, what we call the valley of death there, you can start some biotech development, either in collaboration with a company like we did, or start your own company to the point where you have a formulation and then can start your first-in-man clinical trials. And there's a further funding gate here that is really a second valley of death. And after getting through those clinical trials, there might be a licensing deal with a pharmaceutical company, further clinical trials like the late phase 2 and 3, getting your product to registration. And then once it gets implemented in clinical practice, there may be some evaluating, perhaps other indications for your therapy, adoption, outcomes, and then obviously there's further unmet needs that arise from the limitations of the therapy. So I started here, and my question was really trying to understand rheumatoid arthritis pathogenesis. In fact, I had that question when I went to Dallas, but my mentor quite wisely suggested that I understand basic science before starting to understand disease. So that's what I really set out to do when I got back to Brisbane. And over the years, this question evolved into questions of trying to understand what drove immune tolerance, and then understanding type 1 diabetes pathogenesis, while we were still trying to understand rheumatoid, and it was a bit of the same and some contrast. And then to the real target, which is intercepting disease, autoimmune diseases with immunotherapy. And I've put a black swan, not just because I came from WA, but because of this work that really has been very influential, and it's about anti-fragility. So the term was coined by Nassim Taleb. Anti-fragile is a way of approaching problems and adversity so that you're not just resilient to get through those problems and adversity, but that adversity improves the final outcome. So you can kind of think about COVID-19, and the improved outcome is the enormous array of vaccine platforms that we now have for rapid vaccination technology today. So you might start with an idea or a preliminary result, and then potentially that preliminary result is just noise. So then your research in that early phase is tinkering, required to solve the problems that you come up against and trying to make improvements over time so that your solutions rise above the noise. And as you do that, other people will start to compete with you, and there'll be changes in the environment because other technologies, for example, will shift the goalposts of what you're trying to do. And that, in an anti-fragility setting, increases the likelihood of an unexpectedly big upside. And Taleb and others have coined this the black swan, where most of the swans we look at are white, but then there's suddenly this black swan that actually comes up and hits you in the face, and unless you're looking for it, you won't do anything about it. So the basic science discovery that we made was dendritic cells are a natural component of the immune system, and they present antigens to T cells, the lymphocytes, in the lymphoid organs. And they take up antigens, and they indiscriminately take up antigens because they're phagocytic cells. And they might be apoptotic bodies from dying healthy cells, and they might be pro-inflammatory pathogens such as E. coli. And when dendritic cells take up pathogenic molecules, that activates a key molecule, NF-kappa B, which sets off an inflammatory cascade. And our discovery was that by turning off NF-kappa B, dendritic cells could be changed so that they would silence the immune system rather than activate it. So when you activate it, this will activate T cells. When you silence them, this will regulate T cells. So after making that discovery, the big question that all academics come up against, because your main outcome measure is to publish. And so the key question is, do you patent or do you publish? So the answer is, if you've got something that's important for translation, you patent first. So here I talk to UniQuest, and we patented with our priority date here being August 2002. And then we published in January of the next year the paper, so that we had our priorities in the right order. So then, having patented, it was important to actively pitch my idea to try to get that next round of funding to go towards biotechnology. So I actively pitched, and you can see this was for 6 years, to venture capital firms and pharmaceutical companies. And I did one or more pitches, I counted, to at least 26 different entities around the world, not just in Australia. And the recurring themes, so you get feedback every time we present, even if they don't invest. And the feedback's very, very helpful. So the recurring themes were, we don't like the idea of cell therapy, because our idea was to turn off NF-kappa B and dendritic cells and transfer them with antigen into a patient. So we don't like cell therapy. Can you target to the dendritic cells? And what's your business model for a cell therapy business? Well, no one would ask those questions today, but that's what they were asking in 2004. So we went back to the lab, we listened to the feedback, and my postdoc, Brendan O'Sullivan, and I teamed up with Nigel Davies from the School of Pharmacy at University of Queensland, who was a liposome expert, to develop a dendritic cell targeting strategy in the body, where we didn't have to take them out and put them in. And we did that by marrying, in the figurative sense, a PhD student, Montri Jatrin-Pignot from pharmacy, and a postdoc from immunology in my group, Christelle Capini. And we filed a new patent in 2006, demonstrating that we could use liposomes to suppress via dendritic cells in vivo. So then this is the idea from concept to product. So we've got a liposome here, which is a 100 nanometer nanoparticle, which has a lipid shell that entraps calcitriol. And calcitriol is an NF-kappa B inhibitor. It's also the active form of vitamin D3. And then the aqueous core entraps an autoantigenic peptide, or long peptide or short peptide, and that can be injected subcutaneously, and those liposomes then traffic through the lymphatics to the draining lymph node and target the dendritic cells there that take up the lipophilic particles. And here you can see, and then they interact with T cells there, and because of the NF-kappa B inhibition, the T cells see that antigen in a non-immunogenic or regulatory fashion. And if that antigen is a joint-specific antigen, then those T cells would see the antigen again in the joints, and the regulatory cells would be drawn into the joints. So here's an image from an actual mouse experiment in which we've injected red fluorescent liposomes, and they've gone to the draining lymph nodes within 15 minutes, and you can see them outlining the subcapsular sinus and going down into the lymphatic, and the dendritic cells are labelled here in an aqua colour, and you can see that red starting to be taken up within 15 minutes by the dendritic cells. So in the meantime, the rest of the lab was still engaged in the clinical translation, and we started our first in-human proof of concept dendritic cell trial. So this proof of concept trial was actually using the original idea of making dendritic cells in the test tube, incubating them with an NF-kappa B inhibitor, exposing them to a rheumatoid arthritis peptide, and transferring them into the patient that the dendritic cells were made from. So this started in 2010, after many years of preclinical research, satisfying the TGA with toxicology studies, 3 ethics committees, getting insurance, and then writing a protocol. And at least I knew how to do that. So there were many things I had to learn along the way. So the pivot point for actually translating the rest of the program was in 2010, where at the American College of Rheumatology meeting, I had a poster describing the preclinical development of these tolerising dendritic cells that were about to launch into patients. And at my poster, Michael Elliott came by, and he was the entrepreneur in residence at Janssen R&D in USA. He was actually an Australian that I knew, a rheumatologist who had become very famous because he was the first to deliver TNF inhibitors to patients in London during his postdoc. Now, we talked, and he got interested in the liposomes while we talked at my poster. By 2011, we had finished our clinical trial, and I was invited to present in a plenary session our findings of the liposome clinical trial. And here was the proof of concept. The blood was taken from patients. The blood from monocytes was purified into dendritic cells, cultured with an NF-kappa B inhibitor and cytokines, exposed to 4 citrullinated peptides that were rheumatoid autoantigenic peptides, and those modified cells were injected back into the patients that they came from. The patients, this was a unique trial at the time, patients were selected for high-risk HLA, genotype positive, and autoantibody positive, citrullinated autoantibody positive from all the potential rheumatoid patients that could have been treated. And we followed those patients intensively out to 6 months. So the deal with Janssen occurred after that, and Michael was good to his word, and he brought out the senior leaders, including the worldwide chairman of pharmaceutical research of Janssen, out to Australia, and I pitched, and they liked the pitch and decided to invest. And so how I got there was, as you know, I had done my MBBS and training at UWA, doctorate at UT Southwestern, came to University of Queensland, eventually where I joined became the University of Queensland Diamantina Institute, was supported all the way with Arthritis Queensland, was mentored in the early days by Ian Fraser, and that led me to doing translation. And translation is more than just doing research, which is what I had learnt to do well at University of Texas. It was a fantastic environment for basic scientists. For basic science. But translation, clinicians do this very well because they're trained in this as a doctor. They're trained to manage patients and to manage staff. They're trained in leadership. They might not realize it, but actually this is what they do in managing a whole lot of activities in the clinic. They organize well and they know about products. They understand because they're listening to pharma companies present all the time. They understand in the background about product development. They engage with consumers and they engage with industry. This is something that we all do. So the initial funding that I had in 2006, after we had, and sorry, 2005, as we're about to file our second patent, was from Oz Industry and the Innovation Startup Scheme of University of Queensland. And that's how Dendrite, our spin-off company, was established in 2005. And there are now similar schemes for new ventures, for example, through the MRFF funding. The critical inputs to commercialisation, to actually achieving investment from Janssen, were that I had a drug candidate, which was the liposome. I had a clear patient need, and we had excellent science underpinning it. And we had good corporate governance from UniQuest at University of Queensland, solid patent applications and strategy, and an integrated management team at UniQuest driving industry engagement and negotiating the appropriate deal terms so I could get on with what I had to do and let them do what they had to do. And then I was open and willing to engage with partners. We and Janssen had a mutual desire to help patients. We all focused on the translational goal. I recruited the right people, and they brought a very large team from Janssen to help. And I achieved the milestones along the way towards a tractable product. And that was a long way with Janssen, an 8-year partnership. And I had a very supportive family who really gave me the scope to be able to do this, because you can imagine this is a lot of work, a lot of time after hours, where you're meeting with the US every week or every 2 weeks. And a very good team here at Dendrite from both UniQuest and my CEO. It's a commercial and lab team who worked really, really well together. So what does translation from the bench to the bedside actually involve? We got seed investment and then an options license agreement. We had to do the drug development. We had to think about the lipid shell, whether we were going to have passive or active targeting, which inhibitor we were going to use, which peptide we were going to use, about the pharmacokinetics, pharmacodynamic in preclinical models, the route and dose and how that would translate from mouse to human, the clinical assays for antigen-specific T-cell response that we would do in the patient. Replication studies had to be done from University of Queensland across to Janssen. The GLP and GMP product manufacture, toxicity studies, the protocol again, and then the investigator brochure and the ethics committee. And then so that eventually led to clinical trials, phase 1B, 2017 to 2019. I really had a huge learning and experience from that. I learned new concepts of time, risk, and value. I got an international translational and industry network out of that. And I got the tools and reputation and motivation to do it all again, which is in fact what I did do. So I had a lot of non-industry funding through the NHMRC and continued academic funding after the Valley of Death when we got Janssen funding. And I started to get JDRF funding about type 1 diabetes, trying to understand type 1 diabetes pathogenesis in human from about 2012. And we had a number of grants from JDRF until we got a formulation, liposome formulation for type 1 diabetes. And at that stage, the Helmsley Foundation from the US started to invest, and we've had a couple of grants from JDRF and the Helmsley to develop this liposome formulation, which really has got us over the second Valley of Death for type 1 diabetes. We had a big challenge in 2019 when Janssen pulled their funding after the single ascending dose trial in rheumatoid arthritis because of a big change in management in Janssen. So that's all the data we got for human data from rheumatoid. We didn't get to go on and do multiple dosing. Fortunately, we got all of the SAD data and were able to publish that and take some big learnings from that into type 1 diabetes. So that funding has really got us towards a phase 1B study in type 1 diabetes starting next year. And we'll take all of the learnings that we've got from rheumatoid into type 1 diabetes. In the meantime, we had a partnership deal with Merck for cancer immunotherapy in 2017, and another deal with CSL in Sjogren's syndrome, another autoimmune disease, which started in 2020, which is really seeking to do the same thing and see whether there's a tractable product for translation into the clinic. So how and when did all this happen for me? So here's me with my mum, and then first day of school, graduating from university, first house after we got married, and this is me in my final year of medical school. And then John and I went to the US, and here's me at a conference in San Diego during the 1990 to 97 period. And then starting our family, once we got back to Australia, the research question years, having twins in 2019, in 1999, and then really growing a young family as we started to do that pitching to get funding, and then trying to get through those valleys of death as the children grew. So a lot of fun with the family, and a lot of valleys and troughs and mountains to climb, as you can see here, as we really went through tinkering to try to make those problems more tractable for clinical development. And finally seeing some light at the end of the tunnel as we got that biotech and development, and the second valley of death as we got to the clinic, and children graduating. So you can see that it takes a lot of time and patience to do all of this. And your family does grow up, and you do end up with a bit more time. And to get to the promised land, though, there's still a long way to go through more clinical trials and probably through other indications that will really get to the promised land if we get there. But really keeping that patient in mind, that's really been the driving force to do all of this, because there's a patient at the end. And I continue to see patients in my clinical practice that continue to inform what I do and help me to understand, you know, the landscape in which we're trying to develop these drugs. So if a tolerising approach worked, what would be the impact? So restoring operational tolerance really means re-educating the immune system to remain inert to itself while dealing with invading microorganisms, tumour cells or alloantigens. So it really means safe drugs which allow us to respond still to vaccines and to pathogens and to tumours, so that we can fight those things off while safely tolerising against the autoantigens. So from a patient perspective, this would be prolonged remission off their immunomodulatory or immunosuppressive drugs with minimal intervention and just observation, or perhaps, yearly retreatment, that kind of thing. From an at-risk person's perspective, it would be failure to develop disease with a safe interception to prolonged non-disease onset. And so these would be people like with autoantibodies and some symptoms, but not yet disease. And from the physician perspective, it would be a game changer for clinical practice. It really would change the way clinicians would practice in autoimmune disease. What have I learned? While commercialisation is a long haul and a difficult road, I think you've probably seen that from this talk, it actually is the only way for an idea to get to the clinic and to patients. It just costs too much to do it any other way. And discoveries that never leave your lab do not impact the problems that you set out to solve. So you need to have persistence and the right partners involved. The goalposts and the market needs do shift, and you need to be aware of that and involve consumers to understand that, and pharmaceutical partners. However, as you've seen, prophetic vision is what creates intellectual property. So it means that others won't share your vision for quite a while, but once they catch on, it's a landslide. And now there's a lot of similar technologies around, and there's workarounds and there's competition. But I think, as I've said earlier, you really want to leverage that competition to move faster rather than worry about it. And the key for everybody listening is really, if you have the opportunity to create and translate IP, you can build businesses and you can interact in a way that allow you to translate your technology through to the clinic. So simplify your product, simplify your pharmacodynamic-based biomarkers in a trial design to get answers to fundamental questions and the best bang for your buck in phase 1. You never know when the funding is going to be pulled, so do it as fast as possible. Don't be afraid to look at problems from different angles and involve collaborators and partners with different perspectives. And this will ask you to ask the right allow you to ask the right questions and move forward while learning what does and doesn't work. And innovation is more than just getting a patent long ago. It's actually iterative, and you hope to get more patents because your patents do run out of their lifetime. And I think that's all. I'll stop sharing. I'm happy to take questions.
Thank you. That's an incredible career to summarise in 25 minutes, and it doesn't do you justice. It's an amazing story. You've touched on lots of really important issues there, I think, that are helping people understand what it takes to succeed. Perhaps I could just touch on one of the issues that you raised in your slide. Was about mentoring, and you did mention you had a couple of mentors in your career. Could you just explain to us how that worked for you and what you learned from that?
Actually, I was the deputy director of the Diamantina Institute when Ian was the director, and we would meet weekly. And that weekly meeting, I feel that we shared a lot. So I was reflecting back to Ian as a director what I could see on the ground as the deputy, and the strategy that I I could envisage for the leadership. And he was reflecting back to me as he learned about my journey in commercialisation, and he was mentoring me in commercialisation and was very instrumental in keeping my spirits up, telling me not to give up several times. And I think it's really important to have people with that broader perspective who've been through it, because you always feel that you're the only one going through all these problems. And it's just very helpful to have a broader perspective and to understand that you know, your children will grow and you will have more time. It's not always going to be so difficult. And also helping people to see what's superfluous and that they can give up so that they can actually get more of what they need to get done and not be doing things that are taking up a lot of time, not necessarily helping.
I think that's a great message. I think the message about mentoring, it doesn't have to be a person in your field directly, but someone who's been along the journey ahead of you and can help point out where things are going well, where things might need a change of direction. I echo that 100%. There's a question.
Go ahead.
When you started out in your career, did you expect to work with industry, or was that a bit of a surprise for you?
Complete surprise. I never even expected to be a scientist, to be completely honest. But I was very interested in immunology, and I kept looking for the next challenge when I was a clinician. I kept thinking, oh, it'd be great when I can diagnose patients. It'd be great when I can treat patients. But there just never was enough challenge. So science was great because you never run out of challenge, and industry is just another whole set of challenges. But I love the interface because it's meaningful, it's strategic, it's like a game of chess a lot of the time, and I love games, and I love the personal. So, you know, I'm a people person because that's why I did medicine, but business is all about interacting with people. It's all about, you know, what you can do as a person, bring your competencies to meet theirs. And, you know, I absolutely love that interface. And then influencing my group so that mentoring them so that they can meet the challenges in the time available to actually execute what we need to do. I think it's a great place.
Yeah. So totally unexpected though. So your career takes twists and turns as you go. One last question, because I will sign off now and hand over to Isaac. But a really important one. Did you take time off for your family when your children were young? And if so, how much time did you take off, and how did you negotiate that?
One was 6 weeks. My mum was there. She came from Perth, and literally the week after I got back to work, Sylvie developed meningitis. So it was lucky my mum was there.
Yeah.
Fast. The second was 4 months. I had twins, so it was. I knew it was going to be a challenge to express that much milk, so I breastfed them both for 4 months, then went back to work. And I had a nanny both times, and had a wonderful nanny the second time who could manage the multiples and another daughter. So it was always.
Look, questions are starting to come in now, but look, what I might do is.
Guys, I'm happy to answer or to meet with you.